Hi, I'm Graef from the Center for Bioinformatics of the Universität Hamburg, Germany. In this video, I will present the poster with the title GeoMine, a web-based tool for chemical three-dimensional searching of the PDB. The search for geometric patterns, or in other words, the relative arrangement of atoms, bonds, and interactions of protein binding sites, is computationally demanding but highly useful, for example to comprehend a protein's function. There are only a few tools that address this problem and are focused on protein-ligand interfaces. Also, these are limited in terms of query variability, adjustable search sets, retrieval speed, user-friendliness, and are mostly not publicly available. Our new geometric data mining tool called GeoMine addresses these shortcomings. GeoMine is integrated in the openly accessible Proteins Plus web application suite. It enables searching the entire PDB by using textual, numerical, and geometric queries. The tables for textual and numerical filters are shown here. For example, you can search for complexes that contain a ligand that matches a smart string, filter the PDB title, the PDB resolution, or the pocket properties like the pocket hydrophobicity. All these filters are fully combinable and can be including or excluding to restrict the search to specific complexes. Geometric queries in GeoMine are generated from scratch or with the support of a structure by using a 3D viewer. Next to the 3D viewer, a table lists all elements that are selected in the viewer. Each selected element has a specific color in which it is highlighted. The first possible geometric query elements are the points. A point can either be an arithmetic ring center or an atom. For each point, an element, interaction type, and origin molecule can be chosen. Also, functional groups and the environment by using a smart string can be specified. The second elements displayed below the points are the distances. These can be defined between two selected points and have a length as well as a tolerance range. Like the distances, the interactions are also defined between two selected points. A type is used to differentiate between hydrogen bonds, ionic, metal, pi-cation, pi-pi, and pi-pi parallel interactions. The last possible elements are the angles. These can be defined between distances or interactions, and again come with a tolerance value to enable searching for similar, not exact matches. All in all, these query types give the user full control over the geometric and chemical variability. The results of GeoMine queries are displayed in a list. Usually, the result calculation is done in a few minutes, provided that the queries are reasonably selective. For example, the query shown here results in 59 matches and took about 35 seconds. Each result can be manually toggled to visualize superimpositions of the matches in the 3D viewer. With multiple settings, different visualization styles such as the representation mode for the ligands and proteins can be chosen. By using a download button on the top right of GeoMine, all matched PDB files including a statistics file can be downloaded. Also, further refinement of the current results can be done by clicking the Refine Search button. GeoMine uses the PostgreSQL database technology and is currently about 150 GB in size. This database contains tables that store the textual and numerical properties, as well as the atoms and interactions of the protein-ligand interfaces and an index structure. This index occupies 60 GB of the 150 GB and is based on so-called triangle descriptors. These represent the occurrence of atoms of a specific element, molecule, or interaction type. The side lengths of the triangles are bin distances between 2.5 and 8.5 angstrom. For each of the possible triangles, a bit string is stored. Each of these bits corresponds to a specific point stored in the database. So for example, we have the points marked in purple and blue in the query on the right side of this slide. The arrows between these points show distances between the points and form the triangles. Now because the point with the ID 154 is in the triangle 281, we set the 154th bit in the 281st triangle to 1. This enables us to split a given search query into triangles that can be retrieved from the database in the form of the bit strings, which in turn identify all possible candidate points. GeoMine can be used to search for structural similarities between binding sites of unrelated proteins. This can be helpful in a variety of different applications. For example, it can be used for drug repurposing, to explain side effects, or to discover protein functions. Examples for such applications of GeoMine are presented in our talk and can also be found on the poster. With GeoMine, we present an efficient, flexible, user-friendly, and publicly available web tool for simple as well as complex search queries on the PDB. Almost every spatial arrangement of atoms can be searched, which usually is done within a few minutes. Also, new features, such as the searching of empty binding sites and secondary structure elements, are currently in development.